Hello friends and students. In this video I am going to solve question number 6 of the summer 2020 paper, subject code 9701 and the paper variant 42. In this particular question we have questions from polymerization and organic chemistry, which I found to be particularly challenging, and therefore I decided that I will give a complete solution to this question, because I have got many students asking me to solve this particular question, particularly this part which you see, which is the E part 1 and the part 2, the part 1 and the part 2. This was particularly difficult for the students, and I'm going to show you the complete solution. The earlier parts are not that difficult, but still I will go through the solution. The question says the class of polymers called polycarbonates are made by the reaction of carbonyl dichloride COCl2 with a diol. So you see, this is a diol. Diol means that it has got two OH groups, and this is carbonyl dichloride, and this polymerization takes place in such a way that the hydrogens and the chlorines, they condense to form hydrogen chloride, and this reaction takes place on both the sides of the OH groups, so that this unit can be polymerized. So we have at one end we have an O which will be further polymerized with the other part, and on the other end we have this O joining with this CO, and this unit will be further polymerized. This is how a polycarbonate is made. So deduce the type of polymerization shown here. Obviously it is a condensation polymerization. So now Nalgene is a polycarbonate made from the diol P. So this is your diol, which is actually drawn in this place. In the place of this diol, you have this diol, and it undergoes polymerization in the same way with COCl2, carbonyl dichloride, and we have to form one repeat unit of Nalgene. So following this pattern, you have to these OH groups are actually these OH groups, and this entire part, the rest of the part, is the R part. So taking the clue that this entire thing is the R part and these are the two OH groups, and you have to form a repeat unit like this, you can easily write in place of R, you can write this entire group, and then this O shown here, and then O, and then CO shown here, and then this will be repeated. So this will be the one repeat unit of Nalgene. I think you understood this. So Nalgene is a strong and tough polymer. Identify two types of intermolecular forces that are responsible for these properties of Nalgene. So what are the forces which will be present in the polymer? Obviously London forces, or the temporary dipole induced dipole forces, are always present in all kinds of molecules. So this intermolecular force will anyway be present in the polymer, and the other force, intermolecular force, that will be present will be a dipole, because this carbonyl group is a polar group having a minus charge on oxygen and a positive charge on carbon. So it is a dipole, and therefore there will be dipole-dipole, the permanent dipole-dipole forces present in the polymer. So these are the two forces that will be present in the polymer, which will make it strong and tough. Okay, so I hope you understood this. So we move on to the next part. Proteins are polymers of amino acids. complete the table to show how the secondary and the tertiary structures of proteins are stabilized. So the secondary structure is actually stabilized due to hydrogen bonding, which is present between the C double bond O and the NH group, which is present in the peptide linkage, which is formed between the amino acids in the proteins, and CO and NH are polar groups, and H is bonded to the strongly electronegative element nitrogen, so it is ready to be hydrogen bonded with the hydrogen to the negative oxygen of the carbonyl group, and this is how the hydrogen bonding will take place. And the alkyl groups which are present in the amino acids also form intermolecular forces, and because there are no charges, permanent charges present in the hydrocarbon units, the intermolecular forces will simply be the London forces or the temporary dipole induced dipole types of forces. So these are the forces present in the proteins in their secondary and tertiary structures, which are responsible for stabilizing the protein molecule. Okay, then we go on to this question that explain the significance of hydrogen bonding in DNA in relation to the accurate replication of the genetic information. I think that this topic has not been included in the new syllabus, so it's really not important, but still I'm telling you the answer. You know that there is base pairing, adenine with thymine. These are the nitrogen containing bases present in the DNA, and guanine and cytosine, they also pair up, and the pairing always takes place in this manner because of the hydrogen bonding between these bases, and this is the reason for the accurate replication of the genetic information, because A will always be associated with T and G will always be associated with C. Okay, so state two different processes by which polymers can be degraded. Okay, so if it's a condensation polymer, which can be in the form of an ester or in the form of an amide, then such polymers can be hydrolyzed. The ester and the amide linkage can be broken, but those polymers which are formed by the addition polymerization of alkenes double bonds, and therefore they contain only the single bonds, they cannot be hydrolyzed, but these bonds can only be broken by the UV light, which gives rise to the free radicals. So okay, so now we come to the tough question which I was talking about, and looks you know, this structure looks to be very complicated, and this is the main problem with the students. They are not able to solve this question. So I'm going to simplify this question and tell you the exact solution in a stepwise manner. So this is the cyclic peptide B, and it is broken into its monomers with dilute hydrochloric acid, and one of the amino acids is threonine, which is shown here, and you have to find out the other organic products. So basically you should be very familiar with the hydrolysis reaction with dilute hydrochloric acid, which occurs in a peptide or in an amide. So I have shown here the simplified form of the hydrolysis of an amide. So see, if there is this RONH2, the amide linkage, and if it is hydrolyzed in an acidic medium, then the CN bond breaks, and the CO bond gets attached to the OH, and this is a carboxylic acid group being formed, and the hydrogen adds to the NH2 part, and so this is how you have to hydrolyze it. The CO group will break, the bond between the CO and H will break, and the CO group will change into the carboxylic acid group, and the NH part will add up one hydrogen. So I have tried to visualize in this cyclic peptide the places where the amide linkages are present. So as you can see that if you look at this structure carefully, then you will find that this is the amide linkage CONH, this is the amide linkage. Then once again this is the amide linkage, this is the amide linkage, CONH, this is the amide linkage, this is the amide, and wherever you find the CONH you can see that those are the amide linkages, and then you have to break the CONH bond and attach the OH group to the CO. So if you do this, then you can visualize that this part here will become this part here. If you see this part, which I have shown as glycine, so this will be COOH like this. See this? This will be COOH, and then this will be CH2, and this will be NH2. So this part will separate out as glycine. You can see that very clearly, NH2, CH2, CO, and OH. So this will be our glycine part, and look at this part. So again, since you are breaking the bond here between CO and NH, you will get an OH group here, that is this part will become COOH, and then this is a CH2 and an NH2, right? And then there is a CH2 and the phenyl group. So actually from this part you will get the phenylalanine, which is C6H5, then a CH2, then a CH, then a COOH, and an NH2. So this part will separate out, which is phenylalanine, which is abbreviated as PHE. And so this will be also the phenylalanine part, this will also be the phenylalanine part, and this part, if you see, is once again COOH, and then the CH2 and the NH2, which is once again the glycine part, and this part has been shown separately as the threonine part. This part here is the threonine part. So this is how what is going to happen when this cyclic peptide B undergoes hydrolysis under reflux with dilute hydrochloric acid. So you have to revise the concept of the acidic hydrolysis of the amide linkage or the peptide linkage, whereby you have to create a carboxylic group and an NH2 group wherever there is the amide linkage. And if you apply this concept on this cyclic compound, you will be able to get the fragments in the form of phenylalanine, in the form of glycine, and in the form of threonine. So these are the fragments formed by the acidic hydrolysis. One is the glycine and one is the phenylalanine. And now if you look at the cyclic peptide once again, you can easily see that there is a phenylalanine, glycine, phenylalanine, phenylalanine, glycine, threonine, and phenylalanine. So this is how it looks now. These are the different fragments formed on the hydrolysis of the cyclic peptide. This was a really difficult question to crack, and that's why I used different colors and different bond breakings to show you exactly what are the fragments that are going to be formed when the cyclic peptide undergoes acidic hydrolysis, and hope you have clearly understood. If not, you can refer to this diagram and figure once again, see the fragments which are formed, and use this concept. You will easily understand the various products which will be formed as a result of the acidic hydrolysis of the cyclic peptide. And then how these amino acids can be separated from one another. You are already aware that electrophoresis is one method whereby the different amino acids which differ in their charges and which differ by their masses can be separated when they move to the different electrodes under electrolysis. And the other method is the thin layer or the paper chromatography, where the different amino acids move with different speeds and they get separated on the paper or on the thin layer slide uh, according to their nature, and that's how we can separate these amino acids. So hope you understood the solution that I presented for this question number 6 from the May-June 2020 paper variant 42, and the subject code 9701, which is the advanced level of chemistry. Thanks for watching, and if you liked it, please give it a like and share it with your friends and give your valuable comments. Thanks a lot.